are here in our seminar. We call it Language Research Seminar. So this thing started like 15 years ago with the understanding that what language is, or to discover, or to explore. We were trying to understand Sanskrit in some form. Then we realized through literature, some India statements that the speech is your own, voice is your own. Everyone is endowed with a voice and capacity to speak. Speech is a compilation of information that comes to you through various senses, and it becomes your own interpretation and expression. So is speech. But then the question came, how do you generate speech? What speech is? So then speech has grammar, speech has syntax, speech has sequence. Where do they come from? So we tried to learn some grammar, the Panini, the grammarian, and then we saw that he seems to have discovered that speech has elements. These elements are human elements, possibly anatomical elements. That is, the airflow in the body through various cavities, mouth, nostrils, brain. As they go through, they have their own expressive power. Expressive power means phonetic power. So like any sound mechanism, as things happen through organs, airflow, so different sounds come. He called them syllables, varna, as a word, a Sanskrit word. We call syllable. Syllable means a unit of speech which can be produced. And he said there are 64 of them independent of each other. Nobody has verified what independence is and whether there's a larger set. But because it has been in the intricacies of Sanskrit language and deep literature, it has not been explored. Now what has happened is the same Sanskrit language has proliferated, had gone into spoken by different people, and then the same so-called roots that Panini invented have gone into various languages and create words. So this is the world we live in today. Question comes, the so-called Paninian syllables or the Paninian etymological roots. I won't go into how he discovered the roots. I will say that when I come to my lecture. But these roots, whether they form a foundation of human cognition. That is, as I'm speaking to you, this is, you are getting some sensory information. This sensory information gets converted into electrical currents in your brain, and eventually you create knowledge out of it. So what is the basis of that knowledge making? So basis of the knowledge making are the roots that Panini developed. That is, I can segment this information into these various categories of action. He's saying, okay, I like it, I don't like it, he's too rough, he's too slow, maybe relevant to me, maybe not relevant, I should leave, or it's interesting. All these are different roots of Panini, which are provoked in us by a stimulus. And he said that these roots are 2,000 in number. Again, there's no scientific analysis. Then we come back to say whether what are these roots? Are they innate to the brain? Are there more roots that we don't know? How do roots develop? So these lectures are to explore how we understand. That is, you have an instantaneous reaction to any stimulus. We love with a glance. We appreciate through the first hearing. There's a flash. There is a system of in us. So we call it soul, or we call it mind, we call it mood, but all that is a function of something happening, that our impression is created. That impression is not created in length of time. That impression created instantaneously. So we will try to explore how the stimulus creates impression, and those impressions, how they are carried through into eventually the stimulus causing itself to change the brain state. Brain state in the sense we are absorbed with that emotion, and then there's a time scale to it. So all this in various ways we'll try to explore. So today we have Dr. Bharani Padmanabhan. Bharani Padmanabhan has been here in the US for about 20 years now. He is a MD, PhD from Hungary. Hungary has been very active in neuroscience for a long time. Very able. We have another Hungarian in the team. So Bharani is a neuroscientist. So he will speak to us on the reception of audio signals. So we call the overall series modeling the neural syntax, which basically we are saying that the speech syntax is a manifestation of a neural syntax. So neural syntax is a composition of stimulus segmentation and processing in the neural scheme. So because there's a hierarchy of syntactic operation, so we're trying to create a mathematical model, what we would be saying now, to see whether we can discover how the syntax operates. And hopefully, if we understand the neural syntax, we might understand the language syntax. eventually. So reception, the first talk, reception of audio signals by Dr. Bharani Padmanabhan. The protocol would be like he will speak maybe an hour or so or something like that. And then we have half an hour of or longer depending upon what your questions may be. Listen carefully. And then if you want to join us in your future, we have discussion groups. You can also join there, give your opinion, but ask any question you may have. How do you supplement the line of thinking? So we'll discuss that. So don't interrupt during the talk, then hold your questions and then we will discuss them. And then we'll also set up how the follow-up seminars will go. All right, here is Bharani Padmanabhan, reception of audio signals. Welcome.
Good morning, Suprabhat, Yonabut. So mine is the very, very initial stage of the whole system. It's where the outside signal comes into the human body on its eventual route to the auditory centers where it's understood. So I just realized now on Zoom that I probably should squeeze the slides a little more to the left. Anyway, what is an audio signal? So we have sound waves, they're actual physical waves that physically vibrate the eardrum. And that vibration is transmitted faithfully into the endolymphatic fluid within the cochlear spiral. And that faithful transmission into the cochlear spiral here eventually ends up in this organ called the organ of Corti, where it stimulates these cells, these two different types of cells, and perhaps others as well. And that physical vibration is transformed into an electromagnetic signal there, and it's transmitted out through a nerve. And that's what we now call an audio signal. And so the paradigm for about 80 years or so has been to think of the cochlea as a microphone. So it's a faithful high fidelity microphone, and it just simply transmits audio signals to the brain, to the medulla, and to the brain for actual processing, meaning we don't really know what we're hearing or we don't know that we are hearing until the brain receives the signal. So in the organ of Corti, within the old cochlea, we have about 3,500 inner hair cells and about 12,000 outer hair cells. And they have these hair cells, these cell hairs on the top called cilia that move according to the incoming vibrations. And the classical paradigm focuses on something called polarization, depolarization, hyperpolarization, using ions and spike generation. And it all started with this work, with this paper from 1939 by these two researchers here, Hodgkin and Huxley. So they created this thing that we now think of automatically as a signal. We have a spike wave, and then we have a refractory period. And everybody now knows this because of the electrocardiogram. You know, we have a heart that beats 1 per second, and it shows this sort of signal dynamic. So everybody thinks all signals must look like that. In fact, if you read here, it says, nerve cell enigma solved. And since then, because of the Hodgkin-Huxley model, where they gave an electric shock to a dead giant squid, we automatically assume that ions move in and out of the hair cells, and this generates an ionic action potential in the bipolar neurons that the hair cells each connect to, which then travels through the nerve. And you have that model here. So we have the endolymphatic fluid, which is the cochlear spiral. We have the cilia on top of the hair cell. And then we, following the Hodgkin-Huxley way of thinking, there's depolarization, hyperpolarization, ion channels, ions moving in, ions moving out, and so on. And we, for 80 years now, we think that's the way it does. Here is the terminal bouton of the bipolar neuron, which then goes, it's one neuron, which goes all the way from the hair cell into the medulla. And so we have channels also on top of here, the surface facing the endolymph, and then we also have channels on top of the cilia themselves right there, which you can barely make out here. So we have one neuron per hair cell, and these bipolar neurons are highly specific. So if you take a bipolar neuron from, say, a hair cell that is tuned to the 500 hertz sound, and you grow it in tissue culture and you clone it 1,000 times, each one of those 1,000 clones is going to be a neuron that codes for 500 hertz. And it's going to have that exact length to go into the cochlear spiral to where the 500 hertz hair cell is. So these are very specific, tightly bound connections. These bipolar neurons are also in direct communication with each other, which is a much newer finding. So it's not just a simple pipe or a telephone line, a conduit from a hair cell into the brain. They also talk to each other. And more importantly, they talk to the nerves that come from the outer hair cells. So the nerves from the inner hair cells talk to the nerves from the outer hair cells on the way to the brain. And that's shown here in this picture. And you can see these, tight connections between the bipolar neurons cell bodies. So that's a fairly recent finding, and no one's really quite clear on what they're talking to each other about. So according to the Hodgkin-Huxley model, so we have this picture here of a myelinated nerve heading in this direction from the hair cell to the brain. And we still think in terms of ion channel movement through the axon. And we've come up with this hand-waving idea called saltatory conduction, where the electric current magically jumps from node of Renvier to node of Renvier to node of Renvier in order to explain why it's going through at 100 meters per second. Because if your ion is going through, basically slogging it through the axon, it's not going to make it at 100 meters per second. So basically, we have a model that's not quite useful. So, but for 80 years, that's the model. And if you follow this model, it automatically forces us into thinking that the auditory nerve takes breaks while transmitting the audio signal from the cochlea to the medulla. Sort of like the heart, right? It beats per second, and then it takes a break, and then beats per second, it takes a break. And so, we think, or we've sort of been forced to conclude that decades of research in service of this paradigm, this taking a break model, has not resulted in a full understanding of audio signal reception, because audio signal reception is continuous. It's very important. It's a 24/7 exercise. And we still do not know how we perceive how these hair cells transmit and the nerves transmit in real time all of the constituents of an audio signal needed for survival. So the main theme of our talk here today is survival. So we'll be applying the survival lens to everything and ask the question, will this help us survive? Does this help us survive as a species, as mammals, really? So can you tell the nature of the sound? Is it your mother calling, or is it a tiger roaring? The pitch, the tone, the emotions, is the person being sarcastic? Is it a joke? And what the actual words are? Is it a language that you know? Is it a language that you don't know? All of these things we perceive instantaneously. And so most likely, if you look at it this way, there is no refractory period. So there must be a different way of considering the reception of audio signals. So we are forced to think that there must be something else going on. And we begin by considering first principles. And in our case here, we consider that audio equals survival. The primary function of your ears are to help you survive. Everything else is secondary. Thus, any system that receives audio signals must perforce allow the brain to act or react instantaneously in real time while you can still be saved. So if you apply that principle, there's precious little time for transmitting raw audio to higher neural centers for brain processing, and no time for refractory periods. And that realization, or that new assumption, say, basically underlies the rest of our discussion today. So any new model that we propose, something that doesn't involve ions traveling through axons or hair cells being just passive receivers, must consider that the audio signal is parsed and segmented before the electronic signal is generated within a bipolar neuron, because by that time already precious time has elapsed. And so the cochlear cells, the hair cells, right now we think hair cells, but there are a bunch of other cells in there also, and they must be there for a purpose, though we don't yet know what the purpose is. So somewhere amongst the organ of Corti, amongst the different types of cells in the organ of Corti, we are processing these incoming signals and immediately transmitting the understanding of that signal to the brain through the bipolar neuron. So our main new idea today is that we are segmenting the signal at the initial reception site itself, at the organ of Corti, at the hair cell or supporting cells. So there's the concept distinguishes between receiving the sound and then later understanding the sound, which requires input from memory, which we will go through later. So the zoom pictures on the right side obscure the name of this big round bar, big black bar here, and that's the cortex. So everything is heading towards the cortex. So we have the sound coming into the cochlea. It's transmitted to the medulla. Part of it goes straight to the cortex as pure sound, that I'm hearing this sound, right? Part of it goes to the hippocampus to tell us whether or not we've heard the sound before and what do we think it is. And the hippocampus also involves, perforce, every time the amygdala, which lets us know whether we like the sound in the past or not, or whether it's a dangerous sound and we really should flee. So we've segmented that into these two zones. So there's the receiving zone and the understanding zone. So the receiving zone doesn't require the involvement of memory, but understanding, what Bijoy ji calls knowledge. What am I hearing? That definitely involves memory. So scientifically, there's only one way we can have such a fast response, and that's through the movement of electrons within a protein. And that's not, our discovery by any stretch. We've known that now for 100 years. And proteins, electrons in proteins go really quickly, within femtoseconds of a signal being received. We use this every day when we put a solar cell on our roof. Those are femtosecond electrons moving. So this is what pictures of the atoms look like these days. About 2008, this is what we got. We saw a cloud of electrons around the atom. And then a few years later, they improved a little bit, and we start to see that we have actual orbitals. We have the different higher energy orbitals, and we can actually see them. So these dots don't represent electrons per se, because electrons are both a particle and a wave. So what we are seeing is the probability that somewhere in there is an electron. So these proteins that help electrons move are ubiquitous in both plants and animals. And femtosecond speed electron transfer via proteins is how leaves convert solar energy. And this is what we call photosynthesis. And this is basically how we live. Photosynthesis supports life on this planet. So life on this planet depends on femtosecond electron transfer. And you really don't need expensive nuclear reactors to see quantum physics in action. Any green leaf shows you. So photosynthesis is a quantum electronic activity that involves near instantaneous transfer of electrons via something called quantum tunneling or electron tunneling through proteins with the help of an amino acid called tryptophan, which turns out to be both a unique and extremely important amino acid. It's an essential amino acid for humans, meaning we don't make it, we have to eat it. It comes mostly from milk, and certain other sources, turkey, salmon, and so on, eggs and nuts and whole wheat. But tryptophan is what basically supports life. So here's a wonderful paper from Harry Gray from Caltech who was able to show some years ago already, this was in 2008, that what's happening inside a leaf, every single green leaf, is actually quantum physics. It's quantum tunneling through tryptophan. So what are we talking about when we say energy and quantum? So this is quantum physics, which is not everyday physics. So we have energy levels. A few slides ago, you saw the pictures of the energy levels on the atom. So all molecules and atoms have these energy levels, and sometimes they have a stable conformation, and sometimes they have an unstable conformation, which I apologize, you can't see. But they can switch between those conformations depending on whether or not they kick out an electron or a proton. And if you have this energy barrier, either you have a heck of a lot of energy to surmount this barrier, or you do this real nice trick called quantum tunneling or electron tunneling, where you simply go from B to C, because at that level, you're both a particle and a wave. And that's what we're going to talk about now. It's a consequence of the wave nature. And here's this movie. So in the middle here, there's this barrier, right? It's an energy barrier. So if you were just a particle, you would just bounce right back. But because you're also a wave, you're able to go through the barrier. Yes, the amplitude is different, but you do make some of it through. And that's a whole new concept for most people outside of, quantum physicists. And what's the time that it takes to go through that barrier? It's 1.8 attoseconds, which is 1 billionth of a second. So every green leaf around us does this every single day. Whenever you stick it in the sun, within 1 billionth of a second, it allows electron transfer between proteins. And so we have now the time scale of life. So we have an attosecond, which is what's happening in our cells and our proteins. We have the heartbeat, which is 1 second, which, of course, from a classical mechanics standpoint, is consistent with the Hodgkin-Huxley way of a spike wave and a refractory period, because you have a second. You have time to have refractory periods. And then there's the age of the universe itself, which is a billion. So we are now looking at hearing. All this time we've been looking at hearing as being something around here, but now we are forced to conclude it's somewhere closer to here. And that's what's new today. So we must accept that it is possible that normal physiological processes in eukaryotic cells are also quantum, just like in green leaves. And they cannot be explained by classical mechanics like spike waves and refractory periods. And this has been shown to be true about mitochondria, which are these dynamic organelles which are present in all eukaryotic cells. So this is a little movie about mitochondria. They not only, make ATP, which according to some estimates, we make 50 kilograms of ATP every single day. So we consume 50 kilograms of ATP and make 50 kilograms of ATP, and this is where it's made. But they also have other actions, including regulating stem cell differentiation and initiating apoptosis. So how long a cell actually lives depends on when the mitochondria decides enough's enough. So now we look at the hair cells in the organ of Corti. They have a lot of mitochondria. So they're not just passive receivers. To have that much of mitochondria, and the mitochondria in hair cells have a unique reticular structure. They have a network. They network amongst each other. So they must be doing something. So something's cooking. Something's happening inside the hair cell that we haven't really been paying much attention to. So, we have these very nice micrographs. Later on, I can send people the PDF, and you can actually see the primary sources. And so the new thing today is that we are forced to think that within the hair cells and perhaps the supporting cells, there must be proteins that are doing exactly what the proteins in the green leaves are doing, which is they are receiving energy from outside, which in the case of the hair cell is a physical vibration, and converting it by moving electrons from one protein to another, or from one portion of the protein to another portion of the protein, thereby changing the conformation of the protein. And most likely it's happening by quantum tunneling, because that's the fastest way to move proteins. And it turns out our hair cells are also quite rich in tryptophan. So the tryptophan must be doing something, just like it does in green leaves. So if you don't have tryptophan in your cells, you're going to be deaf. So it's definitely doing something. And the bipolar neurons also need tryptophan because they make serotonin, which is a neurotransmitter. So tryptophan is extremely vital for hearing. So just like green leaves. So you need tryptophan for photosynthesis. Without photosynthesis, we're all dead. And we need tryptophan for hearing. So it's a reasonable inference that it's probably working the same way at the quantum chemical level. So we have the hair cells, right? And we have the nerves going from the hair cells. And then we have all these other guys too. So these are the internal supporting cells, the pillar cells, Dieter cells, Henson cells, Claudia cells, so on. And it turns out they're all talking to each other. They're all talking to each other through something called gap junctions. So they're not talking to each other through nerves. They're talking to each other directly in juxtaposition. And the mitochondria talk to each other within all of these cells. So there's this entire community, this whole network. And if you look at evolution, what does evolution teach? Evolution teaches that if something is there, it's there for a purpose. It's not just simply hanging out, just like the appendix isn't just hanging out, the tonsils aren't just hanging out, they're there for a purpose. And so we still don't know what the full picture is for what all of these other cells are doing, with this new concept that there are proteins and there's quantum tunneling of electrons, and the proteins are the fastest way to convert energy from outside into energy inside. And so they must be doing something. So that's one of the things that we need to think about going forward. Very recently, people have been looking at what's in all of these cells, and it turns out they have a bunch of other different important proteins. Aquaporin 4 immediately catches my eye as a neurologist, because that's the protein channel that's found to be involved in a disease called neuromyelitis optica. So it turns out we have a lot of aquaporin 4 in Henson cells. They must be doing something. This is the tectorial membrane, which for years was thought to be just, like a leaf covering them, a cover, a tarpaulin. Turns out the tectorial membrane is extremely important. It's the one that maintains the calcium levels inside the endolymphatic fluid, and also tunes the frequency for each of the hair cells. So the tectorial membrane above the 500 hertz hair cell is slightly different than the tectorial membrane above the 1000 hertz hair cell. And there are two proteins, tectum A and tectum B, that are mandatory for normal hearing. So if you don't have those two glycoproteins, you're not going to hear. And so these are all very, very new in the last 5 years. Numerous other important proteins have been discovered. Piezo is a very important protein. It's a mechanoreceptor protein. It's one of the ways in which the body transforms physical, mechanical pressure into an electrical signal. Then we have the tippling proteins and numerous other things. And what we don't yet have experimental evidence for is sensory proteins. The proteins that we are postulating must exist as the initial transducers, as the initial receivers of the physical energy, converting it into electronic action. So that needs to be discovered. We think they are probably there, they just haven't been found yet. And what this also means is that they are defined by responsiveness to specific characteristics within the incoming audio signal. So what it means is that the hair cells select for more than just frequency. So it's not just this is a 500 hertz hair cell, that's a 1000 hertz hair cell. There are also proteins that exist to recognize danger, jokes, sarcasm, your mother's voice, and so on. So this is a picture of a grid microarray. We also have these as protein chips or gene chips. But if you think of them as laid out linearly along the cochlear spiral, that's how we visualize our putative proteins. you do this 15,000 times, you get the entire cochlea. So with this way of thinking, the sound signal comes in, changes the electron movement on proteins or in tryptophan near proteins, and this immediate action causes a conformational change in the protein, and that is an integral, intrinsic, innate part of signal transduction. So this is somewhat different from the present idea, which is that the hair cells, the cilia on the top of the hair cell open ion channels, and it depolarizes and creates a spike and a wave and a refractory period. So in the 2023 Physics Prize, which came out just a month ago, the Nobel Committee stated in the world of electrons, changes occur in 1/10 of an attosecond. And we say that that's the level of speed required for survival. And this level of speed requires a different approach to examining the proteins, as I said, to identify the electronic, and I say electronic deliberately, it's not ionic, it's electronic changes that occur within femtoseconds by the movement of electrons. And again, not blaming anyone, we just haven't had the tools to examine this level of speed required for survival. And this lack of tools has also hampered the understanding of audio reception. So it will be necessary to at first computationally model such a system. We can't do it experimentally just yet. That comprises electronic cochlear proteins that change conformation in response to an incoming audio signal. And 100 years ago, people already modeled that for other things. So this is a 1905 picture from Einstein. So this is the photoelectric effect. So we have the photon, which is both in a particle and a wave, hitting a metallic surface, and the energy from this photon kicks an electron off the surface, and that generates a signal. When this electron comes back and joins the atom, it releases that level of energy that took to kick off, and that energy then is converted to electricity, and we have a photovoltaic cell. This is seen in the retina of the eye. So we have a photon absorbed by the 11th cis retinal chromosome, chromatophore rhodopsin. And when the light photon hits, it changes physical conformation to a transretinal. And it actually is associated with a higher energy state, this here. And then so it drops back to a lower energy state and releases an electron. Sorry, I'm going to go through this really quickly because this just shows you the conformation. So it's a physical movement in space. So in our analogy, the sound vibration is the energy that knocks electrons out of a cochlear protein, which has yet to be found. And if you look at all the other senses, if they all follow one way of doing things, it makes sense that all of them follow one way of doing things because that's how you're receiving incoming energy and transforming it. So what we are proposing is that just like in vision, in audition also we have a chemical movement that changes the molecular structure, which is a routine, normal, everyday occurrence in all cells. And this molecular change, this conformational change in a protein must then lead to a fast signaling cascade of electrons that eventually results in an electromagnetic signal that is transmitted to the brain. And this is a complex signal. This is not just 500 hertz sound. It's all sorts of other things in it. It's a 500 hertz sound from a tiger. So very recently, March of this year, so they have found that to be the case in vision, that ultra-fast structural change is the first movement that happens in vision. So if you think of it in terms of sound, this presupposes the existence of specific signatures for specific parameters of sound. And all of this is unknown at this point. What is the sound signature for fear, anxiety, sarcasm, anger, threat to life, love? What are the specific proteins that we have that immediately recognize this in real time? Now, we don't know, but we definitely should think about it. This is already known in terms of visual circuits, not the eye, circuits in the brain, where there are specific neurons that respond to yellow objects compared to red, or contrasting stripes compared to polka dots, or chevrons, or objects moving diagonally, which is different from objects moving from bottom to top. So this is also the proposed mechanism in smell perception. So we have a G protein coupled receptor in the nose that receives energy vibrations from an odorant that causes a conformation change. Energy vibration basically means that the electrons are jiggling, they're moving, the energy is changing the electron conformation, the electron energy state. And there's a lot of controversy in smell about this because for decades now, they've sort of poo-pooed this concept, and they've been going with what's called the lock and key method, wherein the physical shape of an odorant matches a physical shape in the nose. So, you know, a nasty smell has a particular physical shape, and a sweet smell has a particular physical shape. No one's actually proved it. It's very hard to prove one way or the other. But the energy vibrations concept is certainly gaining ground. But again, none of this is as yet known at the level of proteins within auditory hair cells, but it's probably going to happen. So we anticipate that such proteins will eventually be identified in the cochlear cells, maybe hair cells, maybe the rest of them. Tryptophan will likely be the facilitator of electron tunneling because that's what it does everywhere. And this is similar to the established pattern in physics, where they theoretically anticipate findings, you know, theoretical physics. Neutrinos, gravitational waves, all these things were predicted, you know, 60 to 80 years before they were actually found because we just didn't have the tools to find them. But what, you know, we are thinking is that the level of speed required for survival requires a different mode of thinking all the way through, from the hair cell to the brain. And that also makes us think that it cannot be a spike wave in a refractory period in the auditory nerve. And so femtosecond electronic changes require femtosecond speed of nerve transmission. And if you sit and think about your everyday experience, it's true. You hear things instantly, you remember them. You know, it's not as if you hear it and then you have to wait and say, oh, you know, I think it's this, I think it's that. You hear it right away. There's no lag between something happening and you hearing it, unless you're looking at fireworks, you know, many kilometers away. So that requires the transmission of the signal from the hair cell to be transmitted very quickly to the medulla, to the brain. And right now, the fastest way we can think of doing that is as what we call a surface plasmon polariton, which was a new concept to me until this year. But apparently in physics, it's been an established concept for many years. What do I know? I'm a clinical neurologist, so I've been learning. And but it seems to me to be a very viable concept, a 3D surfacing plasmon polariton wave. And that's the key point. It's not an ion traveling through the axon. It's not an ion magically jumping from node of Ranvier to node of Ranvier. It's actually a wave in the axolemmal space. So what's the axolemmal space? So we have a nerve, the axon, and then we have a covering for the nerve called the myelin. And in between the myelin cover and the axon, there's a space, and that's called the axolemmal space, which is very important in this case because it's what you call a dielectric space. The myelin is the dielectric. The axon is like a metal, it's ionic, and that produces the ideal circumstance to create a plasmon wave, a surface plasmon wave that just moves. And it moves very fast. So this is a surface plasmon polariton wave. So the ions you see, they're not really traveling through the axon from left to right. They're vibrating in situ, but altogether, a wave is moving. And so conceptually, this is like shifting from copper wire to fiber optic signaling. And interestingly, 1967, science fiction writers got there already 50 years before us. In Logan's One, they already had optic fibers inside the legs of people. So we think only such a system would provide the speed required for survival. Again, the speed in this situation is different from the speed for a beating heart. So we have to think of a different way. And also provides with a bandwidth. So the difference between regular telephone service and broadband is bandwidth, right? So you can have many, many more signals traveling at the same time, which makes sense if we want to hear everything going on at the same time. Is it a tiger? Is it my mother? Is she angry? Is she cracking a joke? So we have frequency, syllables, tone, pitch, emotion, identity. So we have fast electron signaling in proteins, and then we have fast transmission of those signals as a surface plasmon wave. And so what does it require? It requires us to really shift from the 80 years of the spike wave Hodgkin-Huxley model to thinking of quantum physics, that everything is quantum. It was a revelation to me that photosynthesis is quantum, that, the green leaf in my little money plant down the hall does quantum processes all day. And so does our cell. I haven't spent much time on mitochondria, but our mitochondria are completely quantum. Everything there is quantum. We use quantum physics, quantum mechanics every day to eat, to live, to generate energy. Without quantum, we are dead. So most likely, just like the vision, audio is also a quantum phenomenon, and so we need to start applying quantum tools. And so, we definitely need to sit and model that. There's no other way of actually looking at it in the tissue at this point. We don't know what the proteins are. We don't know where the proteins are. And if you look at the cochlea, it's within the densest bone in the human skull. They're actually very, very hard to get at. But, logically, this is what makes sense. So the real other new thing is that this thing called tagging. So, Bijoyji and Premji are going to talk a lot more about neural syllables. But, we think that to get to neural syllables, we need to first have information tagged by these proteins at the source. So we have these tags from the different proteins sent as an electric or electromagnetic signal, as a wave signal, classifying the incoming signal at entry within femtoseconds, that this is a danger, this is a tsunami, the house is on fire, or that man is shouting at you or something. It's instantaneous. So it's an instantaneous tagging at source, meaning it's not just the sound goes to the brain and the brain processes it and then says, oh, gee, I need to run. And so the tagging also doesn't necessarily always have to do with survival. It also has to do with language. And that's something that we'll be discussing in future. So when the information arrives at the brain centers, yes, already tagged, then it actually makes it easier for the memory centers and the amygdala and then the cortex to transform it into knowledge, to incorporate it into what is already known, and to classify this as this is all new, or I already know this and I know what this is. And from the visual system, we know this to be true. So once again, here on this side here, it says cortex. So there's this thing called Capgras syndrome, where we have the eyes and they send information directly to the cortex. Yes, so we can see. But then they also send the same information to the hippocampus, so we know what we're seeing and to see if we recognize that person or thing. And then we have information to the hippocampus to the amygdala to see whether we like that thing or not. So Capgras syndrome is where a person, mostly after a stroke and sometimes after with a brain tumor, looks at a loved one, a wife, a mother, a child, and says, this person looks exactly like my loved one, walks, talks, speaks exactly like my loved one, but she is not my loved one. This is not my wife. This is an imposter. It's a body double. And the reason for that happening is the eyes tell the person what that person looks like, his wife. The memory says, yes, this is your wife. But then there's a disconnection. It's one of the disconnection syndromes. The connection from the amygdala to the cortex is cut. And so normally when you look at a loved one, that recognition is colored by an emotional color of love, of feeling, of affection, that this is my loved one, this is my wife, this is my mother, this is my child. And we've gotten used to combining all of those at the same time, that when you see your loved one, you expect, without necessarily knowing that you expect it, you expect that color, that feeling to come in at the same time, that rush of emotion that says, oh, my daughter. So that's not going to happen here because there's a disconnection, because there's a stroke. And so what happens is they see that person, oh, this is my wife. She looks like my wife. This very much looks like my wife, but I feel nothing. I don't feel that this is my wife. I don't have that sense of feeling that this is my wife. I feel nothing. Ergo, she's a body double. She's an imposter. And it's a very, it's not that rare. We see it. I've had 2 patients with it. So basically, the receiving section is fine. The knowledge and understanding section is somewhat impaired. So it's logical to infer that something similar also happens on the hearing side. 60 years ago, on the visual side still, they showed that there was a difference in terms of knowledge between lab-bred monkeys and the wild monkeys. So the lab-bred monkeys didn't really intentionally avoid live snakes because they just didn't know what it was. I mean, they all saw it. I actually found an old video, and
issue like this one have no reason to fear snakes. Their counterparts in the wild, however, will have learned to fear snakes from watching the reactions of other members in their group.
So knowledge is different from seeing. And so it's logical that the hearing system operates similarly. So we have our fundamental principle is that the function of the brain is to protect the individual. And you see that in numerous ways, in terms of temperature, in terms of sight, in terms of smell, and of course, you think in terms of hearing as well. And the whole purpose of the system is primarily to know that there's an immediate danger from someone, a fire, a predator, poisonous mushroom. So the new thing that we are talking now is that we visualize proteins in the organ of Corti as the sentinels, the gatekeepers, the guardians, and that they work very similarly to the fast proteins in leaves, fast proteins in the receptors of the eye. It's more likely than not that the process of hearing, receiving the audio signal, is quantum, and so we have to apply quantum tools. So it's just a simple inference at this point, but it's probably more real than the Hodgkin-Huxley model. Thank you.
Can I ask all people to unmute and have a round of applause for Bharani? Please unmute. So now, 1, 2, 3.
Thank you. Thank you, G. Thank you very much.
All right. Okay, so what you heard is the first talk in our series. Now, something I should have said as a part of what you saw, or part of the motivation that we had. So that is, the brain goes into these states, emotional states. And these emotional states are psychological states as done by the scientists in the mental field. But they have been called Rasha states in Indian literature. That is, this particular stimulus that you were injected last hour did create a state in you. Is not a state, a combination of states. So the question is, those are real. You are being affected not by the signal, but your interpretation of the signal. So that we said actually we wrote a paper on it, from the Russia theory, we stated that the brain may have these 8 fundamental states which manifest. What that might mean is, so these states happen, as Bharani said, pretty quickly. Disgust, love, fear, 8 states are there. So they happen right away. And then probably they evolve, they moderate themselves, they go into subsequent states. So the metamorphosis, all this happens in course of time. So the question then would be, from Bharani's point of view, that how the states are detected. Are they detected? So detection is quick. So he thinks it's quantum mechanical, but proteins are not found yet, or we think proteins exist. So anyway, with that basis, that there are 8 fundamental psychological states that we're all governed by, which eventually manifest themselves. We have Professor Shastri with us. He knows more about this, about the Shastra, about like how these 8 states. Now, 8 states are not the only states. There may be many other ancillary states, but they don't manifest with that signature. So when we get morose, depression, then the state lingers for a long time. We do not really hear properly. Or when you are in love, you can hear more. Then, so those kind of things. So now I will open it up. So let us have discussion. Feel free to ask questions to Bharani on what he proposed. He said lesser than what the overall scope of the work is, but he tried to be articulate on this quantum mechanical thing that he has lately been thinking about, that instantaneous recognition or transmission to the hippocampus that something is happening and you must alert yourself. Then the question is, one, that initial communication takes place, whether the brain processes afterwards are guided by that initial intimation, which will kind of say that how you perceive music, psychophysics of them, there's a tune, that something happened. So you went into a state, and that state then let reinforce more material, more neurocenters got engaged. So you have now an absorption on it, an immersion on the state. So that process, which we think is physical, we have to understand scientifically. So please be free, ask as you like, and discuss. We have time, and let us see, help us to understand. I mean, this is all exploratory, and so.
I have a simple question, Bijoy.
Yes.
That's an excellent talk. I can understand.
Introduce yourself. I think so the people know your background.
So I'm a theoretical physics, condensed matter theorist.
Yeah.
And I have looked at the problem of electron transfer in proteins and a long time back. And I think everything is electronic. And the question about time scale is relevant. But I have a simple, we are talking about acoustic timescales, frequencies, which are, of course, much slower compared to electronic time, femtosecond timescales. But I have a question. Your whole theme was survival. Everything nature has done, information is stored or created with some survival as a biological process. Does this mean when the sound comes, you have initial receptors, proteins, and so on? Of course, eventually brain has to be involved. But is there any local storage of information which the brain doesn't have to do, or it does automatically there, so that brain doesn't have to think about survival and so on? Some memory locally in those proteins which are connected to the hair cells.
We have no idea. So in the nervous system, we do have some reflexes that are integrated in the spinal cord itself. So, we have the edge reflex, the touch reflex. I mean, if you touch something hot, the spinal cord takes care of it instantly. You don't have to go all the way to the brain and say, oh, this is hot, it's going to burn my skin. But since we haven't really been thinking about sound as a fast quantum process, there's been no research, or we haven't known start about it. So that's a wonderful, wonderful idea. In terms of survival, is it handled locally? Whereas if it's love and sarcasm and jokes, is it transmitted up? And who makes that decision? I don't know if we're going to be able to find the answer for a while. I don't know if it's also true, because I don't think the brain ever completely delegates anything and keeps away, because I think the brain likes to be directly involved in everything. There's no such thing as an autonomic nervous system. The brain knows exactly what the autonomic nervous system is doing. So there may be an instantaneous pre-programmed response locally, but I don't think memory is local.
Good. Yeah. Well, information is an important part. Yeah. So go ahead. Yeah.
No, what I was saying was the amount of current. The question is the way we are planning to model. We have started some work, is to see how much metastable, how much energy we need to knock off electron from a metastable protein. How many are there? Do they exist? And then how much energy we need in order to do that. And then what happens is, so in the acoustic, whether as the packet being created, as the sound is received by us, is there a component in it, in the sound, as I'm speaking to you? So that is let's say pitch, that's tone, intensity, but there's a component in it, naturally, universally, which is connecting you to this sound. So that component, which is a scientific component, which is a part of this, when you make a convolution of this frequency signal and so on, which will connect to the so-called protein, and then say that that is fear, that's anxiety, that's love. So what has not happened is the signal has been interpreted as sound signals. But sound signals also have life in them. So living sound signal has characteristics then. And you as a living receptor also have to handshake with this living signal. So that part, how does that handshaking happen? So now, philosophically or in other matter, religiously, people have called mind, mood, something like that, but we have to quantify them, saying that they are in the brain, is happening. And so that's the goal. And that's the I said before, so-called Shakti. So that is, so we have to, again, we're writing it, we're not published yet. So we shall see. But it's a great question. I think so join us. I think the question is the protein knocking. This thing came like a month ago, and the knocking people have been talking about for a long time differently. But this metastable, now the question then could be what is the alternate theory? Why do psychophysics has statistical theories? So statistical theories are not working in life. And he used a very strong word, survival, but survival is a very global word, larger word. Survival has evolution to it, but it's like a stroke function. It's like a gate. It's good for me, not good for me. Do I quit? So like that survival, is necessary, not necessary. So like that. So you have taken that particular yes-no operation. So that yes-no operation is probably, it doesn't happen in test, or doesn't happen in order, it doesn't happen in sight. You can't escape them. But in the sound, it lingers. It can affect you. The neural syntax can be decomposed by virtue of bad words. And then you get immersed in it. You can go into depression. So that we have to understand. How does this process take place? Sound in emotion. There's a lot of literature in India, this thing in a literary way, in a poetic way. But and also I did some experiment with Valmiki Ramayana, and it happens. The words can put you in a mood. So what do words do in the mood? See how the words composed in particular manner and recited in particular manner can transcend to you to create a mood.
State of your mind, yeah.
So how does that happen? So the question is, so that's not even though it happens in an evolutionary way, it must somehow catch on it to develop it. So that's a new gentleman, a Hungarian scientist called Bucsaki. He's kind of coming up with theories. He's in Colombia about how does the brain operate. So the question then is, it seems that from his point of view, so there are innate neural centers built in our brain, which are expecting signals to arrive. So it's not the signal is processed, but the tools of processing signal are there in the brain. So that is, but then the question is, what are they? What are they means they are not simply chemical compositions. What is the knowledge value of them? So that is where we are trying to think when is the language syntax develops, which eventually the expressive part of the brain, whether the proto part of it, the origin of it, may lie in the neural centers. So that means my statement, as it comes to you, it gets segmented into thousands of parts, and then each part is checked whether what emotion it has, and that emotion gets transmitted to a neural center to process, because that is pre-wired. Neural center knows from knowledge point of view that he is talking this. So that science of it, we have to, and then we shall see when that neural centers then will be reinforced, and they will grow. Neural clusters will form. And that we are checking through to see whether, as we speak, so how many syllables we can say at a time. What is our conceptual limitation? How do you form a concept? There's a boundary to it. So there's the electronic stability to it. That is, you can't really, you have to break it there because that neural cluster is not stable anymore. So you can form so much, but then you compound. You compound from one to the other with different actions. So that becomes Panini. How do you really?
That's my later concept, yeah.
And then the other part is the syntax. Syntax, that is where we are trying to do. That is, all this suppose even happened. How does it make a sentence? So that individual neural centers now have been reinforced and grown, and they're clusters. So who is that monitor? Who will then say, okay, this is how knowledge is. This is how the streamline is. A neural sentences. So is it electrical? Is it electromechanical, electrochemical? So we shall see. So that's the plan of this whole effort.
I think it's very, very, yeah.
So we shall see how much it's another way to look at it. And any other question? Anybody has?
Jadev has a question.
Yeah, please do.
Thanks, Dr. Padmanabhan. It's a very thought-provoking talk.
There is some background noise. Sorry, someone is talking. Sorry.
Who is?
No, I think, yeah. So you can mute now. People can mute now.
I have a simple question here. That in a movie, when we see that there are 24 frames per second, gives you a sense of continuity. So that tells us the sensitivity of our system. Basically, that brain cannot recognize the gap between two frames if they are running 24, which means it is, and if the electrical signals are running in a period of a millisecond. So if we compare the electrical signal that is coming from the audible, from the hearing system to the brain is running at a millisecond, and the visual system cannot go beyond 1/20th of a signal or part of a second. So if we look at this way, is it not possible that hearing system can also not recognize, even if there are some gaps? As you said that there are some gaps, it's not a continuous signal. So even if suppose it's not a continuous signal, but that gap is very short, is it possible that the hearing system can work based on the old model? I'm not particularly in any particular favor of model, but I'm simply trying to see what is the thinking there.
So 24 frames per second is the minimum required for flicker-free vision.
Yeah.
It doesn't mean that we cannot see faster than 24 frames per second.
Okay.
So but that's the minimum required. In fact, when you don't have to see a movie, when you're just looking around every day, we have gaps. We have gaps 60 times a second. Every time you move your eye,
yes,
your visual field switches off and switches on again. And we are trained to not notice it, right? When you move your eyes left, when you move your eyes right, because we are not just a camera with a film, right? We are an electronic sensor. And so the electronic information is processed and you're generating this image in the back of your head. And when you move your eyes, you're not generating an image during that movement. And so it's blank. And in certain strokes, people can perceive those blanks, and it's very disconcerting. So yes, that is a wonderful thing. What is the minimum refractory period that will go unrecognized? And the answer is we don't know. But it's certainly much shorter than the refractory period of an electrocardiogram. And it's certainly much shorter than the refractory period that we're currently seeing with the giant squid axon. So we don't necessarily need to have a femtosecond wave. I fully agree. In fact, we don't know what the speed of these polariton wave is going to be because no one's tested it. There's one bunch of researchers, Dr. Jacek and Dr. Jacek in Wrocław in Poland, who are the first to actually propose this model of a plasma and polariton wave to justify how you have faster nerve transmission in myelinated neurons compared to unmyelinated neurons. And there's one group in London at University College, I think, that actually applied it to unmyelinated neurons also. And in both cases, they showed that this ion moving through the axon business just doesn't hold true. And so, the refractory period, there may be, so, Bijoy used this word packets. So waves may also be packets. So you could have a packet wave, and then you have another packet wave, and the gap between the packet waves, we don't know what the ideal gap would be. There probably should be some gap to allow for the nervous system to send the stuff from a logistical perspective. You go to this nerve center, you go to that nerve center, right? Because those packets probably come with those tags, and something is reading those tags, probably in the medulla, and saying this tag goes to the amygdala, that tag goes to the hippocampus, this tag goes to the uncinate fasciculus. This tag goes straight to the auditory cortex because it's just a pure sound signal with no other information associated with it. All of this is unknown at this point, but that's what we are now starting to actually think about.
Right.
If you ask the question,
to distinguish between, I think, what he's talking about, impression versus processing.
No, he's talking about speed.
Right. Impression is quick, immediate. So what impression does is, what we think is creating a neurocenter network through the impression. This is what it is, the tiger. So that impression is kind of communicated. So then the next part of it, you can really reinforce because you have to set up the stage. So neurocenters have to be arranged, alerted, and then you can do whichever way you are trying to make the processing to take place, slow, fast, and this thing, but you have created the mood, the impression, the framework. So that we think is Paninian in the sense that is some kind of a impulse in us through a scene, through a stimulus, that this is fire. So the fire then causes itself in some kind of a Paninian activity, which is neural. And then the question is, how big is the fire? Is it coming at me? Is it this? Is it this? Is it this? So cognition, and then recognition. So cognition, the process most likely happens. So let us see. We are all wondering. We're just exploring because there's a lot of different things are coming into it.
Just one, the time scale for that process, what you call instantaneous concept of the whole fire or this, cannot be faster than femtoseconds or the time scale of global response.
We don't know.
You're talking about a global response and has to be a longer time scale. It may look instantaneous from a different point of view, but if I were doing a local electronic measurement, that process has not come. So what you are talking about, global states, time scale of global processes.
Glance, like for example, a glance, I do not know. You are a good point. So this whole thing developed from this so-called how much information a glance has. Like suppose you reach Jupiter, the object reached Jupiter. So it has the first glance of this landscape. So then you infer information from that glance. It is the instantaneous image. It is not the processed image. You don't know there is no air. You don't know many other things. So that glance, whether that, Bharani calls survival. So that is a life in you talking to the object outside, the stimulus outside, to connect. Am I in the right place? This is what we think. Can I take it in? Do I see more? Do I clear my bandwidth to receive more information? Do I escape? So that may have, what we also have thought that maybe some other reaction take place, muscular, vascular, eye dilation might be also taking place.
It's worth thinking about. I mean, there's something called entanglement in quantum mechanics. It is almost instantaneous compared to physical. So maybe those states you're talking about are some sort of entangled states.
Yes.
Which communicate much faster than
Yes.
the physical response and local memory. Those are real series of physical processes. So
That's
the states,
exactly, yeah.
But anyway,
we're trying
this
to
is
understand.
very fascinating.
Okay, one more question here. Where you see? Sibaram had a question.
Can I, sir? Yes, yes, sir.
Okay.
Namaste, sir. Couple of questions. Sorry if I'm doing a leap of enthusiasm actually here in asking this question. Question number one is related to that interesting insight on the importance of mitochondria in this, in the processing of that information where the signal is received. Okay, so is there any extended research being done in terms of, say, because mitochondrial DNA from a genetic perspective comes from mother, so is there anything done in that context of auditory signal processing and the mitochondrial DNA from genetics? That's my question one.
Not yet.
Not yet. Okay. Okay, sir. And my question two is around the tryptophan, because tryptophan got me very excited because my imagination ran wild because tryptophan is one of the primary source of food. Tryptophan is from cow's milk. And when I look back to the Vedic culture, where the memory, sound, speech is their Vedic culture of all the mantras, the Shruti culture, they obviously had a lot of cows around. I'm sure their cow milk must have been much more better than what we have today. So is this that the tryptophan and audit signal processing can be linked to, supplementing tryptophan, say through milk or any supplementary, anything around that, sir?
So the tryptophan comes from food. It's an essential vitamin, meaning we can't make it ourselves. So all tryptophan comes from food, depending on what your food source is, and most of it comes from milk. So there's no way around it. It's definitely dietary.
So by doing diet supplementation, we can improvise that auditory processing.
It's way too early to say that, because all we have now is that we do see a lot of tryptophan in hair cells. That's all we know at this point. Tryptophan supplementation in the late 80s caused serious illness called Guillain-Barré syndrome. And tryptophan supplements were actually banned in the United States between, I think, 1989 and 2004. For that reason, because the quality control just wasn't there. But there's absolutely nothing wrong with you drinking milk.
Thank you, sir. Thank you.
Well, Professor Shastri, are you around, or you want to have any comments? Venkat Shastri? Yoga Sanskritam? Not there. All right. Anybody else has any? Devnaran, do you have any comments?
Yes. Padmanabhan is a very thought-provoking talk. A lot of things resonated in me. By the way, I'm a physicist and a semiconductor device engineer, or power electronics and normal electronics. I think in addition to quantum mechanics, you have to bring in relativity, theory of relativity. That means the velocity of light is constant in any material. C is equal to nu lambda. That means the frequency and wavelength are related. Okay? That means the waves that you are going to need to explain depends upon the dimensions of the system that you are investigating. So I have a question about the quantum tunneling that you are talking about, the attosecond. It appears to me that the potential that you are talking about is dynamic. It's a function of both space and time. Is that right? Way of looking at it?
By space, you mean within the cell, within the protein?
Yeah.
Oh, yes, it has to be. But because we don't know which protein it is, I can't tell you how much space there is. In photosynthesis, it's in the level of angstroms. So in the human cells, probably about the same.
Okay. And the other thing that occurs to me is we are within matter. We are talking about like excitons or polaritons or plasmons. That's because they are sort of combinations or pseudo states of electron and light. Okay. By light, I mean not visible light, but electromagnetic waves. The point I'm making is the electromagnetic waves in the universe is everywhere, right? Whereas the polaritons or plasmons that we are talking about is within our cells. But there is an inherent connectivity between these two. Because as I said, the polariton is a combination of electromagnetic wave and mechanical wave or electrons and things like that, right? So has somebody looked into these broad aspects of connectivity, the whole universal connectivity with us? That means
you mean like using a near field communication device to listen in on your nerves?
I mean, when you bring in near field, I think what I'm saying is that the near field and the far field are connected. That's what I mean. Yeah, you're right.
But go ahead. But no one's done any of it because even this plasmon polariton as the means of nerve transmission is very new, like 2022 new. And other than these two poles in Wroclaw, no one else is even talking about it. I somehow found it because I went looking for an alternative to the Hodgkin-Huxley model.
I see. No, I think I've been thinking of getting into medicine. As a matter of fact, there was a surgeon I met in Chicago airport, and he said, Deva, you should really be working in the medical field. Because a lot of work needed.
Different discussion.
Needed to understand what is happening in the heart. For example, you are saying heart signal.
The only thing I can comment is the following.
So I think that I like your speech. I mean, whatever you said, and I may be interested to go back to understand the interaction from a fundamental point of view. That means it's basically a field and matter interaction. Thank you very much for your talk.
You're very
Professor Sastry, are you around? Do you want to speak something?
Yes, can you hear me?
Yes, we can hear you.
Thank you. I'm not able to start the camera. I don't know what technical glitch.
So Professor Sastry is a yoga teacher, and he has been doing Sanskrit for a long time. And he has been interested in this neural connections. He is in connection with me for about 5-7 years now. So he is gracefully attending this lecture. So let us see how he sees whatever we are presenting to him. B.V.K. Sastry, you are from Bangalore, is it?
Yeah, I am right now in Bangalore.
Yeah, okay, go ahead.
Okay. See, one of the things I'm struggling to understand with different branches of science, whether it is the neuroscience or the biology or the vision science, whichever we call, is up to a certain point, the neural imprints, the chemical imprints, the process go on into the brain, the physical, manageable, observable brain. But when it has to move beyond that to whatever space called the mind, where the rest of the things are processed, how does it switch over? So you are explaining several of the experiences in the language of the material and the chemistry and the processes. The information in the classical text uses a different language of the physiological parameters, technically what we call as the nadi. So the understanding of the traditional nadi concept or the biomaterial concepts as saptadhatus and other things to the modern physics, physiological, biological language is extremely challenging. Can you help me to understand what you have done on these things? So when you use a new name like the chemical, what you are mentioning, it becomes difficult for me to map in my mind to which part of the traditional understanding I should go. Memory of the sound. Okay, let's say the k sound memory is different from the memory of, let's say, the p sound. A lot of explanation is given, but how do you map it in the modern context?
So that's something that Premji and Bijoyji are going to be working on, the actual neural mapping. So today's talk was basically about how the sound gets to the head.
See, nadi, it could be, we have not really gone to that point yet, but I think I can probably comment on our larger picture of what we are thinking, that the human cognition is quantized. Human cognition has independent channels. So those initial channels are universal. Universal means, as we talked before just now, so there's a connection to the universe, to the brain. That means universe creates a signal, any signal, visual, olfactory, audio, doesn't matter, but brain can process it. So brain processes it through these emotion channels. This is the hypothesis. So that is a gateway there. So this gateway, as today's lecture, Bharani said, could be proteins, which are the gatekeepers of the universe to the brain. Now the question is, as I said, Bujsaki and so on, what the brain has besides these gatekeepers. And that is where your nadi may come in. There are anatomical locations where signal would be cleared, in the sense which is again part of the Bharani's lingua of survival. So that is the particular emotion protein which is engineering on the corti, the vibrations. So then it communicates to a neurocenter. So those neurocenters in the brain, they are organized through vascular connections to your nadis. Now, nadi, the architecture of them, what they do neurally, is something we have not studied yet. Maybe you can educate us in the process. But the question, what we have been trying to think is that how does this stimulus, as it comes from outside, how does it become knowledge for the individual? And so nadi, from that point of view, is a defense as a reaction to it, but probably not in the processing part of it. That's what I would think, in the sense that once I create a understanding of it in the Bharani's map, reception and understanding. So once the understood, then I react. And that reaction is whether my leg or hand or heart or but you can educate us on the Nadi Shastra, which you have not really. I think Prem did certain pictures of it long time ago about how the nadis may operate and so on, but I don't think we can claim that we have studied it to some depth to understand the defense part of it. So my point is we have three or four maybe: reception, processing, recognition, then defense. But
can I just share my understanding at this point? Now, I understand the details of that. I've been in discussion with several of these supposed senior persons, and the basic question that's being discussed is, I see a red flower. What is the input that's coming as the visual input, and where is that redness issue? So they have brought in, the scientists have brought in, right from Leibniz, the cogito and all that, and then to the signal and the brain process, the neuron triggers, chemistry, all sorts of things they have brought in. Now my question has been very simple. If I have a clear memory, a conscious thought, understanding of coffee experience is different from tea experience, can you explain me the difference between the two? How do I make a choice between the two? The detail of the experiment was very simple. The other way around is, suppose in your that experimental cage, you have got a set of mice, and there are two roads to go. On one end you have got the cheese, and on the other end you have got the nuts, and you just open the gate. By instinct, some go towards the cheese and some go towards the nuts. Can we understand what is that neuronal chemistry that triggered this particular mouse to go to one side and filter out the other one? So
that we are exactly trying to explore. I think this series of 4 lectures that we are creating is exactly engineered to answer such questions. What is the signal is a brute signal. But signal has a quality in it. He mentioned mother's voice, tiger, sarcasm, which is same thing as coffee and tea. So the question then is, how much of our neural framework is designed to detect parts of the signal at what time? What we are saying today is that is an instantaneous. For example, coffee and tea are probably not distinguished. But what is distinguished at the beginning, that it is drinkable, looks hot and so on, I'll be careful. Something of that nature, which is very femtosecond operation, is a very immediate glancing operation. Then you process it. Then the process goes through. So then you say, this is not what I needed. This is coffee. I don't drink coffee. You throw it. Or this is, I take it. So those kind of operations do take place as gradually. Then what happens is that these information with the initial input by the femto, hot liquid, something stimulant, something of that nature. So that in the neurocenters creates a syntax, which is, this is coffee, which I don't like, I never drank, I only drink Uti coffee, this came from England, all that comes as a sentence. So this is what we are thinking, the neural processing of this signal. And then what you do is, so that distinction, object distinction, that this is the coffee I drank in Malabar Hills 10 years ago, that happens in much later in the process. So we have to stagger it. But the question, what we have been thinking quantitatively is, so there is a discretized operation that the brain can bring into knowledge chain, and that knowledge chain is there. And Bharani used the word tag, memory tag, which means this tag goes through, and eventually that tag connects to other tags in the memory where to bring information, and the information blob from your basic signal has become a larger blob of, so which becomes a neural sentence. What we may say, we have not, that how that suppose sentence creates, then how do you compose it? What the syntax of it? Left to right, right to left, high potential, low potential, chemical, protein, who controls it? That we don't know yet, but we have to somehow engineer Panini to it to see that the hierarchy of this, there's a progression of it, there's a basicness of it. That is true or not, we do not know, but we have to experiment it computationally. That what comes first is my mother's, when I drank that first cup of coffee, did it remind me of my mother, or did it remind me of the coffee shop that I went in college? So that kind of thing. What is the hierarchy? Who controls the hierarchy, if at all? So we shall see. It's all exploration. Do join and contribute. What we'll learn. I don't think we have any set ideas. We have simply a problem. We simply enunciate a problem that this is because the linguistics in the world is not thinking about this. Cognition as a human operation. The cognition is language operation. That is, you are told it's a tiger, hence it's a tiger. So as though that if you are not told, you don't know what tiger is. So that's, we think, is not correct. So, okay, with that, so let us do join. I'll invite you when we have next meeting.
Okay, so before
and let Prem,
step out of there, okay, I'm glad you mentioned about Panini, and I'm still focused upon the Shiva Sutras, the 14 of them with the 44 basic units and processor clustered. The way the AI people and the neurolinguists and semiotic people, the pharmacologists, biologists, everyone, they're studying this character set, or the Varnakshara Samamnai, technically what we call as the set sequence cluster of the sounds as a social language entity units. Somehow, I am strongly tend to believe that these are systemic properties of the human system. You call it the brain or the mind or whatever it is at the very highest level. So, as a subset of these sounds, we get the social language, which in other words means if I were to type my name in English, should I use SASTRY or SHASTRY or SASTRI? It is a process, but somewhere deep in the mind or the brain, whichever place it is, the sounds are clear. It is a sh sound, then a long after that, then s, then th combined, the re combined, and re short like this. So that doesn't change anywhere in the world. We were right.
We have not come that far yet, and I don't think we can come that far at all. What we are lately, maybe, again, from my point of view, coming to that possibly each letter, each varna that you talked about, it has its own universe of knowledge. It is a blob. It's a large quantity of human information. It contains in it. That means the universal information, knowledge, is contained, segment of it, in one of those. Now the question then would be, is there a hierarchy of it? So is there a potential difference between a da and a ka? And then what a da is? But a da is a purely breathing instrument, or what we say is a cognitive element, but it may not be da itself. It may be a proto, small segments of it, which are probably like figments of syllables, which are these neuro segments. So they may be combining to create a da and a ra or a pa or something. So that we'll see whether we can model. But you have to give us time. We are not there yet. We are simply starting up to understand it. And we may not be in the right, hopefully in the right direction, but that is the model we're trying to, in the sense that there's no pre-frozen da or a pa or a ba, but there are neural segments which are pre-frozen. So much we know. But we don't know what those neural segments will translate to. We know they have information. So we're trying to see what that information is. We do not know even now how many neural centers exist. So that is the model is there are innate neural centers. They do something, they communicate, and they have neural waves. So they're in phase, and then this transmission takes place. Then they have a handshake. Then the neural center connects to other neural centers, and possibly through small segments of communication, then the Paninian burden forms. That is, this is a concept of evil. This is a concept of love. As you say, concept of red, concept of flower. So that develops through those neural connections. So flower is not a neural center. Flowers is segmented to much smaller parts to become a neural. This is our thinking. But we do not know how to segment it, and then what is the basic element of segmentation. But that we'll try to invent through model to create it. So since we're getting late, let me now, Prem, who's our next speaker. I think the date is November 18th. So we will go into, let him articulate what he plans to do. And then we'll meet in, and then hopefully we'll discuss further. Okay, Prem.
Yeah, so thank you, Bijoyji. I must say to Bharani that it was an eloquent and very convincing presentation. And your thesis that audio signal carries a protein marker is a start point for us, and for my session. So the session on 18th November is reading the neural centers. And in our audio signal, which reaches to the auditory cortex, it carries the protein marker, as Bharani presented, which implies that it also carries its energy and it reinforces the associated neural centers. Now, labeled with the emotions, a marker which should help make the connections in the neurocenters network, where the proteins are distributed or assembled in a distinct space, would be a question. However, the audio signal becomes a neural circuit through a process where it gets segmented into 30,000 auditory nerves, which travels through the electronic process, which Bernie explained. And it activates a neuronal ensemble that transforms into perceptual units. Now, this perceptual unit then gives us some kind of coherent perceptual experience, and which includes the neural circuits, the connectivity, the segmentation, perceptual, and the organizing the sensory information. Now, coming to the neural syntax, a pattern of neural activity and organization and encoding, which isolate the features and incoming auditory signal through hippocampus and amygdala. A lot of emotional and complexities are reinforced through the auditory cortex. The neural circuits gives us the connectivity to the neurocortex and the ensembles of neurons that encode specific aspects of the memory and spatial information. The read neurons are part of these circuits, serving as an intermediary for the information transfer. Now, how this entire process gives us the understanding of what the object is, and how do we then subsequently perceive it, would be the thrust in the session which I would be having on 18th.
Good.
Thank you.
Bela, you want to comment anything at all? Is Bela around?
Yeah, Bela should be around.
You're asking me?
No, Bela,
Bela.
our friend. He's also part of our, I think. Bela, are you there? He's not there. Okay, all right. So why not we unmute ourselves and then thank all people. So Bela is gone. So we'll reconvene on November 18th. So thank you all for participating. And Professor Sastry, do join us and educate us as we proceed forward. Sibaram, you're always welcome. Dimnan, welcome. And we shall see. All right. Okay. Until November 18th. Happy holidays.
Namaste.
Yeah.
Okay.
Namaskar.
Durga Puja. Durga Puja.